Modern pharmaceutical companies have spent billions of dollars attempting to synthesize compounds that Amazon tribes have been extracting from jungle plants for thousands of years. They have not succeeded. Not fully. Not in the ways that matter most. The substances these tribes produce in clay pots over open fires, using knowledge transmitted entirely through memory, continue to do things in the human body that laboratory-produced alternatives cannot replicate. This is not folklore. This is the conclusion of peer-reviewed research published in journals that do not traffic in romanticism. The first thing to understand about psychoactive and medicinal plant use in Amazon tribes is that the Western category of drugs does not apply. The word carries assumptions: recreational use, altered states pursued for pleasure, substances existing outside the structure of ordinary life. None of those assumptions accurately describe what these communities do with these plants or why they do it. Among the Shipibo-Conibo people of the Ucayali River Basin in Peru, the preparation and administration of ayahuasca is not a casual event. It is not available to anyone who requests it. It is controlled entirely by the curandero, the community's trained plant specialist, whose preparation for that role typically spans years of dietary restriction, isolation, and supervised apprenticeship before they are permitted to prepare the brew independently. The ceremony in which it is administered is structured, specific, and embedded in a framework of intention and accountability that has no equivalent in recreational drug culture. Ayahuasca itself is a combination of two plants whose interaction produces effects that neither plant produces alone. The vine Banisteriopsis caapi contains compounds called beta-carbolines that inhibit a specific enzyme in the human digestive system. The leaves of Psychotria viridis contain dimethyltryptamine, DMT, a compound that the inhibited enzyme would normally break down before it reached the brain. Together, they produce an experience that lasts between 4 and 8 hours and has been described by researchers, clinicians, and participants across cultures in terms consistent enough to constitute a documented phenomenology. The question that stopped Western researchers when they first encountered ayahuasca was not what it did. It was how Amazon tribes knew to combine those two specific plants out of the 40,000 species present in the Amazon ecosystem. The beta-carboline inhibitor and the DMT source had to be combined. Either plant alone produces no significant psychoactive effect. The combination requires knowledge of a biochemical interaction that Western science did not formally describe until the 20th century. When researchers asked tribal elders how this knowledge was first acquired, the answer given consistently across communities was that the plants themselves communicated it. That answer has been dismissed, debated, and reconsidered by anthropologists for decades. What has not been dismissed is the result. The combination works. It has worked for longer than any written record of the Amazon exists, and the people who use it did not discover it by accident. Curare is the substance that most directly changed the trajectory of modern medicine, and most people who benefit from that change have never heard of it. The word curare refers to a category of plant-based preparations used by multiple Amazon tribes as a hunting poison applied to blowgun darts and arrow tips. Its active compounds, primarily tubocurarine, derived from the plant Chondrodendron tomentosum, cause rapid muscle paralysis in the target animal by blocking the receptors that allow nerve signals to reach muscle tissue. An animal struck by a curare-tipped dart loses motor function within minutes and dies from respiratory failure as the paralysis reaches the muscles controlling breathing. What makes curare medically significant is the same property that makes it effective as a hunting tool. Controlled muscle paralysis, delivered at the right dose, is one of the most valuable capabilities in surgical medicine. Before curare-derived compounds entered Western medicine in the 1940s, surgeons performing abdominal operations had to use dangerously high doses of general anesthetic to achieve sufficient muscle relaxation for the procedure. The margin between the dose that relaxed muscles and the dose that stopped the heart was narrow enough to kill patients regularly. Tubocurarine, isolated from curare and introduced into surgical practice by Canadian physician Harold Griffith in 1942, changed that calculus entirely. It allowed surgeons to achieve deep muscle relaxation with dramatically lower doses of general anesthetic, reducing operative mortality and expanding the range of procedures that could be performed safely. It became a foundational component of modern anesthesia protocols and remained so for decades. The Amazon tribes who developed curare did not know any of this. They knew that the preparation paralyzed animals. They knew the specific plants required, the specific preparation process, which involves boiling the plant material through multiple reductions over many hours, and the specific application methods that made it effective as a hunting tool while remaining safe for the hunter to handle and for the meat of the hunted animal to be eaten afterward. That last point is critical and frequently overlooked. Curare is orally inactive. It must enter the bloodstream directly to have effect. The tribes who used it understood this distinction functionally, even without the biochemical explanation, because they observed it across generations of use. The preparation of curare is not simple. It is not a matter of crushing a plant and applying the result. Different tribes across the Amazon developed different formulations using different plant combinations, each producing slightly different paralytic profiles, suited to different hunting contexts. The Waorani of Ecuador used formulations optimized for speed of onset, critical for hunting fast-moving prey in open canopy. The Matses of the Javari Valley used formulations with longer onset times but deeper paralytic effect, suited to larger animals encountered in denser vegetation. These are not accidental variations. They are the result of deliberate refinement across generations towards specific functional outcomes. Western pharmaceutical researchers attempting to synthesize tubocurarine and its derivatives in laboratory conditions produced compounds with similar but not identical effects. The naturally derived compound continued to outperform synthetic alternatives in specific applications for years after synthesis became possible, because the natural preparation contains a profile of related compounds whose interactions the laboratory versions did not fully replicate. Cambo is the substance that most consistently produces discomfort in Western researchers attempting to categorize it, because it does not fit neatly into any existing pharmaceutical category, and its documented effects challenge assumptions about what plant-derived compounds can do in the human body. The preparation comes from the skin secretion of Philomedusa bicolor, a tree frog found throughout the northwestern Amazon. The Matses people, who are among the most documented users of Cambo, apply it through small burns made on the surface of the skin. The secretion enters the body through the burned tissue rather than through ingestion. What follows is an intense and predictable physiological response lasting between 20 and 40 minutes that includes vomiting, sweating, swelling of the face, and a dramatic drop in blood pressure followed by a period of elevated alertness and physical energy that users describe as lasting for days. The pharmacological profile of Kambo is unlike anything else in the documented natural world. The skin secretion contains a complex mixture of peptides, short chains of amino acids, that interact with the human body in ways that Western pharmacology spent decades trying to fully characterize. Among the compounds identified are phyllocyaurulin, which affects blood pressure and smooth muscle contraction, phylomeduzin, which affects mucus secretion and smooth muscle, and dermorphin and deltorphin, which are opioid peptides with a binding affinity to opioid receptors several times greater than morphine. That last point arrested the attention of the pharmaceutical industry when it was first documented. Dermorphin and deltorphin are among the most potent naturally occurring opioid compounds ever identified. They are also structurally unusual. They contain amino acids in a form that does not occur naturally in mammals, which is part of why they interact with human receptors so powerfully and why synthetic replication of their full effect profile has proven difficult. The Matses do not use Kambo as a treatment for pain. They use it as a preparation for hunting, specifically for the period of heightened sensory acuity and physical endurance they report, following the initial acute response. Anthropologist Peter Gorman, who documented Kambo use among the Matses in the 1980s and was among the first Westerners to undergo the ritual, described the post-response state as a period of extraordinary physical capability, reduced fatigue, heightened senses, reduced hunger that lasted well beyond what the acute pharmacological window could account for. Researchers have not fully explained this. The compounds identified in Kambo's secretion account for the acute response, what produces the extended functional enhancement that Matses hunters report, and that Gorman documented in his own experience, remains an area of active investigation. The possibility that the secretion contains additional compounds not yet identified, or that the interaction between identified compounds produces emergent effects not predictable from any single compound alone, has not been ruled out. What is documented is that the Matses have been using this preparation for hunting for generations, that they have developed a precise protocol for its administration that minimizes risk while maximizing the functional outcome they seek, and that Western pharmacology encountered this preparation and found it contained things it had never seen before. Rapé, pronounced ha-pai, is perhaps the least discussed of the major plant preparations used by Amazon tribes, which is significant because it is among the most widely used across the broadest range of communities. It is a powder, not a liquid. It is made from a combination of finely ground tobacco, Nicotiana rustica, a species with nicotine concentration several times higher than commercial tobacco, and ash from the bark of specific trees, combined with additional plant materials that vary by community and by intended use. It is administered through a small pipe blown directly into each nostril by another person, typically a shaman or senior community member, in a precise and forceful insufflation that delivers the preparation deep into the nasal passage. The immediate effect is intense. The high-concentration nicotine enters the bloodstream through the nasal mucosa rapidly, producing effects that are pharmacologically distinct from smoked tobacco: faster onset, higher peak concentration, different metabolic profile. The ash component, which varies by preparation, contributes alkalizing compounds that affect how the nicotine and other plant alkaloids are absorbed. The additional plant materials in ceremonial preparations have been documented to include species with stimulant, analgesic, and, in some preparations, mildly psychoactive properties. Among the Huniquin people of the Brazilian and Peruvian Amazon, rapi is not used recreationally. It is used at specific moments, before hunting, before ceremony, during healing sessions, and as a component of the ayahuasca preparation process. Different formulations are prepared for different purposes, with the selection and preparation remaining the responsibility of specialists whose knowledge of the specific plant combinations and their effects is among the most carefully guarded in the community. What connects ayahuasca, curare, kambo, and rapé, what connects all of the major plant preparations developed by Amazon tribes, is a principle that Western pharmacology has only recently begun to formally articulate. The principle is that biological systems respond differently to complex natural preparations than they do to isolated purified compounds. The presence of multiple interacting compounds in a natural preparation produces effects that cannot be predicted from studying any single compound in isolation. The interaction itself is pharmacologically active in ways that single compound synthetic alternatives do not replicate. This principle has a name in contemporary pharmacology. It is called the entourage effect. It was formally proposed in the context of cannabis research in 1998 by Israeli researchers Shimon Ben Shabbat and Raphael Mechoulam. Amazon tribes did not have a name for it. They had something more useful: 4,000 years of empirical observation of preparations that exploited it consistently and deliberately. The pharmaceutical industry's difficulty in fully replicating the effects of these preparations is not a failure of chemistry. It is a consequence of attempting to isolate and reproduce individual components of systems that were designed, through generations of human observation and refinement, to work as systems. The curare formulation that outperformed early synthetic tubocurarine did so because it contained a profile of related compounds whose interactions the synthesis did not capture. The ayahuasca combination that produces its characteristic effects does so through a biochemical interaction that requires both components in their natural form. The kambo secretion contains peptide interactions that no laboratory has yet fully characterized. In each case, the Amazon tribe got there first, not through luck, not through instinct, through the same process that produced every other body of knowledge these communities carry. Sustained attention to what happens when specific things are done in specific ways, accumulated across generations, refined by consequence, and preserved with the same care that any civilization reserves for its most valuable knowledge. The jungle gave them a laboratory. Time gave them the patience to use it properly. What they produced in that laboratory, modern science is still working to fully understand